Hi, everyone. Thank you so much for joining this session. We are going to discuss a very important topic, which is mechanisms of antimicrobial resistance, both bacterial and fungal. So without any further delay, I would like to invite our speaker for this topic, Dr. Anusha Gobinathan, who is the head of microbiology laboratory and quality manager of Molecular Diagnostic Center at SRM Medical College Hospital and Research Center, Chennai. She also plays a very important role in the infection prevention and control, as well as the antimicrobial stewardship program at the hospital. She has a total of 32 national and international publications, 3 patent and 1 patent grant to her credit. So thank you so much, ma'am, for accepting our invitation and for joining this session. Over to you.
Thank you so much, Vrunda. Dear students, so today we'll be dealing with a very important topic, which is antimicrobial resistance mechanisms. And please feel free to give me your questions or feedback at the end of it. So it will be very useful for me. So this is my introductory slide. So I'm a professor at SRM Medical College. And today, this next 40 minutes, we will be concentrating on mechanisms of antimicrobial resistance. So we'll deal both with bacteria as well as fungi. So first, we need to understand what is antimicrobial resistance before we dwell into the mechanisms of antimicrobial resistance. Now, there are various terminologies that we have to understand. One is called as persistence, and this is called resistance. Now, what is persistence? Persistence is the bacterial cells which are not susceptible to a drug, but they don't have any resistance genes. Okay, so the drug does not act on them. It does not function. It does not kill the bacteria, but the bacteria does not have any resistance genes for that particular drug. So this is called as persistence. This is not resistance. Now, resistance happens when a bacteria, which was initially susceptible, slowly becomes resistant to an antimicrobial agent. So in this case, when the bacteria propagates, its daughter cells would all be resistant. This is called as genetic transmission. Difference between persistence and resistance. Okay, so persister cells, when it propagates, the daughter cells needed to be resistant to the drug. While as a resistant cell, when it multiplies, the daughter cells will be resistant to the drug. Okay. Now, what is biological resistance and clinical resistance? Resistance refers to the observable reduced susceptibility of an organism to a particular drug, to a particular antibiotic. While as clinical resistance is when antimicrobial susceptibility, that is the sensitivity of the bacteria to a particular drug, has been lost, and this drug can no longer be used for any clinical indication for the patient. Okay. And there is another important terminology, that is intrinsic and acquired resistance. Now, intrinsic resistance is something which is inborn in the bacteria. Okay, and so these are drugs which you don't have to check using antimicrobial susceptibility testing in the microbiology lab, because it will always be resistant to that particular bacteria. So it is intrinsically, like for example, by birth of the bacteria, it is resistant to that particular drug. While as acquired resistance means, initially the bacteria was susceptible to the antibiotic. Later on, it has developed some change. Okay, either it can be in its gene plasmids or any mutations, which has caused the bacteria to lose its susceptibility. Often, what we find is that overuse and misuse of antimicrobial agent is the single most important cause for developing this particular acquired resistance. And also, there is a terminology called as selective pressure. So, what a selective pressure is? If one particular antibiotic is being used, okay, so slowly what will happen is the bacteria which are introduced to this particular antibiotic will slowly start develop resistance to that particular antibiotic. Now, for example, if you had 100 bacterial cells and you have used penicillin for a long time, so this penicillin will kill, might be say 96% of them. The remaining 4 bacteria, okay, which are resistant, will be able to thrive, and when they multiply, they in turn will produce a resistant population. So this has happened selective of the antibiotic. Now, what are the factors which favor the spread of antimicrobial resistance? The most important factor which favors the spread of antimicrobial resistance is irrational use of antibiotics. Okay, that is using antibiotics left and right, no matter what the clinical condition of the patient is. The second thing is uncontrolled sale of the antibiotics by over-the-counter prescription, without prescription. So, for example, just for a cold or for a throat pain, which could also be because of a viral infection, people go to the pharmacy and get antibiotics over the counter without any prescription. Number 3 is poor infection control practices in the hospital. So because of this, what happens is, if there is any resistant population of bacteria in the hospital, it will thrive and it will cause hospital-acquired infection to the patient. Number 4 is inadequate sanitary conditions. And number 5 is inappropriate food handling. You would not believe that resistant population of bacteria are also introduced to patients through food. Okay. So your eating habits will also contribute what kind of population is there in your, what kind of bacterial population is there in your gut. Okay. So these are the factors which favor the spread of antimicrobial resistance. So this is intrinsic resistance, which I told you in a nutshell before. It is the innate ability, okay, or you can think as it is an inborn ability for a bacteria to resist a particular group of antimicrobial agent. Okay, so if you see the diagram, okay, the bacteria is always drug resistant to that particular antibiotic. So it is intrinsically resistant. Well, as a second picture, you can see that initially the bacteria could kill, initially the drug could kill the bacteria, but later on, the bacteria has developed certain adaptations and it has acquired resistance against that particular drug. Okay, so this is the difference between intrinsic resistance and acquired resistance. Right. So some of the examples for intrinsic resistance is a Klebsiella bacteria being intrinsically resistant to ampicillin. Okay. Enterobacterial, that is E. coli. So your gut bacteria, majority of them will fall under enterobacterials. They being resistant to gram-positive coverage of antibiotics like clindamycin, vancomycin, etc. Okay. A Citrobacter being resistant to first and second generation cephalosporins. Pseudomonas being resistant to tigecycline and cotrimoxazole and ertapenem. Acinetobacter being resistant to fosfomycin, aztreonam, etc. So whenever we see a antimicrobial susceptibility report from a microbiology lab, you will see that certain drugs will not be reported for certain bacteria. For example, ampicillin will not be reported for Klebsiella. That is because the lab will not check ampicillin for Klebsiella because it is intrinsically resistant. Okay. And same way for Pseudomonas, nobody will issue a antimicrobial susceptibility report with tigecycline because tigecycline is inherently intrinsically resistant. Okay, so these things we should have in mind as a microbiologist. We have in mind when we are releasing the report. Also, the clinicians or the clinical pharmacists, when they see the report, they should understand why these drugs are not being reported. Okay. So these are some gram-positive bacteria which, okay. For example, Enterococcus is resistant to aminoglycosides, that is amikacin and gentamicin, to cephalosporins. So you will not see these drugs being present in the antimicrobial susceptibility report of a patient. Okay. Right. Now, resistance can be transferred by mutation or it can be transferred by transferable drug resistance. These are two very common ways of transferring the resistance. So these are the acquired form of resistance. Okay. So resistance occurs because of mutation. So certain genes are undergoing mutation. Whereas transferable drug resistance occurs because of plasmid transfer. Okay. So this plasmid can be transferred through various mechanisms. It could be transduction, transformation, or conjugation. So it doesn't matter. So basically there what happens is a plasmid is getting transferred from one bacteria to another. And this plasmid has the drug resistant genes. That is why when the plasmid gets transferred, the recipient bacteria will also become resistant. Okay. A mutational drug resistance will generally be a low degree resistance. So a resistance to one drug at a time is caused by mutational drug resistance, whereas a transferable drug resistance will be a high degree resistance. Multiple drug resistance can be transferred at one particular time. A mutational drug resistance can be overcome by combination for tuberculosis. They give a combination of 4 drugs in the line panel. Whereas if it's a transferable drug resistance, you cannot overcome that using drug combinations. A mutant drug resistance of the resistant mutants are generally low, but here the virulence does not alter. The virulence of the bacteria does not, and the resistance is not transferable in the case of mutational drug resistance, but in the case of transferable drug resistance, the resistance is transferable. Okay, so that is because you have plasmids which will transfer from one bacteria to another. horizontal spread, while as mutational drug resistance, it will happen only from the parent bacteria to the daughter cell. So it is a vertical spread. So this is a difference between mutational drug resistance and transferable drug resistance. Okay. Now coming to the topic of today, that is mechanisms of antimicrobial resistance. So you have multiple mechanisms which causes antimicrobial resistance. We'll first start with bacteria, okay? And this is a diagram which shows you the most common types of mechanisms of antimicrobial resistance. The first is impermeable cell membrane or cell wall, okay? The second thing is modification of the drug target. The third one is inactivation of the drug. And fourth one is pumping out of the drug using efflux pumps. So these are the very most common mechanisms of antimicrobial resistance which you will see in bacteria. Okay. Right. So the same thing. So decrease permeability across the cell wall, efflux pumps, enzymatic degradation of the particular antibiotic, and modifying the target site of the antibiotic. So these are the ways in which an antibiotic loses its power to act on the bacteria, right? So we will start one by one. The first thing is decrease permeability across cell walls. Now what happens here is usually the drug will enter inside a bacteria through the porine channels. Now what happens is the bacteria will modify their cell membrane porine channels, and this will in turn prevent the antimicrobials from entering into the cell. Okay, so this is called as decreased permeability across the cell wall. So coming to the porine channels, there are two main ways in which the porine channels will undergo changes. Now, first thing is when there is a decrease in the number of porins, and number two is when porine channel. So because of these two changes, permeability of antibiotic into the cell through the, for example, bacterials, that is, it comes under the Enterobacterial family. They carve up in group of drugs, that is your meropenem, because of reduction in the number of porins. Okay, that is the first mechanism. Okay, so you can see in the diagram a diagrammatic representation of the drug not being able to enter into the bacterial cell. Next is biofilm formation. Now, what happens is the bacteria, certain bacteria have the ability to produce biofilm. Now, this biofilm will protect the bacteria from the attack of the host immune system. Okay. Also, it will protect the bacteria from attack of antimicrobial agents. Okay. So, the most commonest bacteria which produces biofilm is Pseudomonas. So, biofilm is a very effective mechanism to prevent the entry of antibiotic into the cell. And what is biofilm? Biofilm is nothing but a slimy, sticky consistency material which is made up of polysaccharides, proteins, and DNA of the resident bacteria. Okay. So, because of this biofilm, the antimicrobial agents are not able to enter into the bacteria. The next mechanism is the efflux pump. Now, what does an efflux pump do? Efflux pump, as the name suggests, it will pump the antibiotic from inside the bacteria. Okay, so it will mediate expulsion of the drug from the cell, and these are actually transporters. These efflux pumps are nothing but they are cell membrane bacteria. There are 5 families of efflux. You have the AT binding cassette family, the multi-drug compound extrusion family, the small multi-drug family, the major facilitator super family, and the resistant nodulation cell division family. So these are the various efflux pumps which are seen most commonly in bacteria. Okay. Now, the ATP binding cassette family, it is found in Vibrio bacteria. So, the Vibrio is a bacteria which is known watery diarrhea. And this is able to efflux fluoroquinolones, that is your ciprofloxacin, okay, fluoroquinolones and tetracycline from within the bacteria. The second type of efflux pump is your multi-drug and toxic compound extrusion family. Now, this can cause efflux of fluoroquinolone, the same ciprofloxacin, and some aminoglycosides. Okay. Now, the small multi-drug resistance family efflux pump. These are hydrophobic in nature. Okay. Now, what they will do is they are seen in Staphylococcus species and in E. coli, and they can efflect antibiotics such as ampicillin, erythromycin, and tetracycline in Staphylococcus. And in the case of E. coli, they can efflux vancomycin, erythromycin, and tetracycline. Okay. Now, there is the next family, which is called as a major facilitator superfamily. Now, this is present in the case of Acinetobacter baumannii, which can pump out erythromycin and chloramphenicol. E. coli also has this pump, which can pump out macrolides, fluoroquinolone, and trimethoprim. So, 50% of the efflux pumps in E. coli are this major facilitator superfamily efflux pumps. The resistance nodulation cell division family efflux pump is seen in Pseudomonas aeruginosa. Okay, so this mainly causes Pseudomonas to develop resistance to anti-Pseudomonad agents. The third type of antimicrobial resistance mechanism is enzymatic degradation. Okay, now this is commonly seen in the case of aminoglycosides. Now, what happens here is the bacteria will produce enzymes called as aminoglycoside modification enzymes. Okay, some of the examples of this is N-acetyl transferases, phosphotransferases, adenyl transferases, etc. So, this will enzymatically degrade the aminoglycoside antibiotic. So your aminoglycoside antibiotic is nothing but your gentamicin and amikacin. Okay, right. And your beta-lactam group of beta-lactamases, okay, those enzymes, okay, which are very, what I say, it is a hot topic these days in antimicrobial stewardship and in antimicrobial resistance. They also cause enzymatic degradation of beta-lactam antibiotics. Now, what are beta-lactam antibiotics? Any antibiotic which has the beta-lactam ring structure, okay, that is called as a beta-lactam antibiotic. Okay, so what this beta-lactamases will do is it will hydrolyze a particular site in the beta-lactam ring structure and thereby cause degradation of the antibiotic. Right. So beta-lactamases, we will again see it in detail a little bit later. Now, they can be classified based on Ambler's classification or Bush-Jacobi classification, and they will have either a serine residue or a methyl ion. Okay. Right. So this is the beta-lactamases Ambler classification. So you have class A, B, C, and D. In class A, if you see, you will have your ESBL enzymes. Okay, you will have your CTX enzyme, TEM, SHV enzymes. Then you will have your KPC enzyme, NMC enzyme. Okay, so all these enzymes, your KPC and your NMC, all these things will cause resistance against carbapenem. So they are carbapinamases. Now coming to Ambler class B. Okay, in Ambler class B, you have the WIM, IMP, the most favorite NDM. Okay, so again, these are carbapinamases. In Ambler class C, you have the AC carbapinamase. In Ambler class D, you have the OXA 48 and OXA 23. Okay, so this is how beta lactamases are classified. So all these beta lactamases, basically what they are doing is they are causing the degradation of the beta lactam drugs. Okay. Next is modifying the target site. Now, what happens is in certain bacteria like MRSA, that is methicillin resistant Staphylococcus aureus, okay, the target site of penicillin is the penicillin binding protein. Now, in these bacteria, what happens? The target site is altered into PBP2A. In some instances, PBP2C. So then the antibiotic cannot act on the cell wall. Similar thing happens in Bacterium tuberculosis, where streptomycin resistance is caused because of modification of 16S RNA. And in rifampicin resistance, there are mutations in your RNA polymerase. So basically, the target sites are getting modified and thereby the antibiotic is not being able to act on the bacteria. So in the case of VRE, that is vancomycin resistant enterococcus, okay, your VAN genes are present which are responsible for causing resistance against vancomycin. What will the VAN genes do? They will cause changes in the peptidoglycan structure, okay. So that causes decreased binding ability of vancomycin. So again, the target site is getting modified here. Then coming to daptomycin, okay. So daptomycin is a higher antibiotic used in gram-positive bacteria. Now, daptomycin requires calcium for binding into the cell membrane. But when there is mutations in the MPRF gene, now what happens is there is a change in the cell membrane surface. This prevents the binding of calcium and therefore it prevents the binding of daptomycin. So all these things are example for antimicrobial resistance caused by modification of the target site. Okay, right. Now, for example, if you take a gram-negative bacteria like Pseudomonas aeruginosa, there are multiple ways it can have resistance. It can develop resistance. So it has intrinsic resistance, it has acquired resistance, it has adaptive resistance. So if you see intrinsic resistance, the presence of efflux pumps, okay, the presence of having a low outer membrane permeability, and presence of having a antibiotic inactivating enzyme, all these things are a part of causing intrinsic resistance, okay, towards antibiotics. Whereas if you see acquired resistance, it develops these resistance by mutation or by horizontal gene transfer, that is through plasmids. Okay, and what is this adaptive resistance? Adaptive resistance occurs because of environmental factors. It also occurs because of continuous exposure to antibiotics. Okay, so these are various mechanisms by which one bacteria can develop resistance. Now, I would like to detail about beta-lactamases. So we already saw the classification of beta-lactamases. We saw the Ambler classification of beta-lactamases. So in that, we saw a term called as ESBL. Now, ESBL stands for Extended Spectrum Beta-lactamases. Now, what does this mean? If a bacteria has ESBL, that means it is resistant to all penicillins and first generation, second generation, third generation cephalosporin, that is until ceftriaxone, ceftazidime, everything will be resistant. Okay, cefazolin, cefoxitin, cephalothin, everything is resistant, and monobactums, they are also resistant. Okay, and here what we have to know is ESBLs will be killed by carbapenems. They can also be killed by ceftriaxone. Okay, and they can also be killed by beta-lactamase inhibitor. For example, amoxiclav. So that can also kill amoxicillin clavulinate. That is a beta-lactamase inhibitor. So that can kill an ESBL. Okay, so an ESBL is resistant to penicillin first, second, and third generation and monobactams, but it is sensitive to carbapenems, cefamycins, and BL-BLI combinations. Coming to AMSA is ESB plus. Okay, they are resistant to statins. But here, what happens is they can be killed by carbapenems. Okay, but BL-BLI will not work on AMC. Okay, on ESBL, BL-BLI will work, but on AMC, BL-BLI will not work. Right? So AMC is nothing but ESBL plus it is resistant to cefamycin plus it is resistant to BL-BLI. Now there are a lot of methods we identify AMC in the lab. Just I thought out of input some 2 slides. Okay, so one is your phenylboronic acid, wherein on a cefoxidin disc we will add phenylboronic acid, and this is a lawn culture of a bacteria. Okay, a lawn culture of a bacteria on a Muller Hinton agar, and then we'll put 2 discs. One is a cefoxidin disc, another is a cefoxidin disc plus phenylboronic acid. Now, on this combination, if you see an increase in the zone size of the bacteria, okay. So if you see a zone size, you know the bacteria is being killed by that particular antibiotic combination. So if the zone size is more than 5 millimeter than compared to only cefoxidin, that means this particular bacteria is AMC positive. Okay. The methods also cefoxycillin double disc synergy method. Okay, so in one end we will keep only one drug, in the other end we will keep a combination. Okay, cefoxidin and cloxacillin combination, and we will see the increase in the zone size. Right. Now about AMC, we saw about ESBL. The third thing is carbapenemases. Now in carbapenemases, what happens is, I told you AMC can be killed by carbapenem. But in carbapenemases, they are resistant to carbapenem. Okay. In carbapenemases, there are 2 major types. One is the serine type, and another thing is the metallo-beta-lactamase. Okay, which has the metal ion. So again, there are various methods to identify carbapenemase in the lab. There is the MSIM method, the Carba-NP method. Okay, so these will not be of much use to you. So I'm not going to dwell into it. Okay, so this is just a picture of Carba-NP, where by color change in the tubes, we can find out whether the carbapenemase enzyme is present in the bacteria. Okay, so if it changes color from red to yellow, we'll be able to find that carbapenemase enzyme is present. So the drug choice for the resistant pathogens. Okay, so this is a small picture which gives you a nutshell idea of what antibiotic you can use for resistant bacteria. For example, if it's an MRSA, a methicillin resistant Staphylococcus aureus, you will find that most prescriptions, okay, will have vancomycin, linezolid, etc. If it's a systemic infection, okay. If it's a simple infection, it can also be cotrimoxazole or clindamycin. But if it's an ICU patient, most likely you will see vancomycin and linezolid. Now coming to ESBL, if the person is having ESBL, you will see in the prescription 4th generation cephalosporin, or you will see carbapenem. If it's ESBL produced by an enterobacterial group of bacteria, okay, that is E. coli, Klebsiella, etc., you will see that the prescription will be for BL-BLI, okay, BL-BLI combination, your piperacillin tazobactam, cefoperazone sulbactam, all these things are BL-BLI combination, or you will see a carbapenem prescription. Now, in the case of AMC beta-lactamase producers, okay, the clinician will write for carbapenems. Okay, and in the case of vancomycin resistant enterococcus, generally you will see linezolid, daptomycin. Okay, you will see this combination drugs like minoprestin, dalfopristin, etc. Fosfomycin in case of UTI. So these drugs will be used in the resistant enterococci questions. In the case of Pseudomonas, okay, you people generally prefer to use piperacillin tazobactam and aminoglycoside combination, or carbapenem, fluoroquinolones, and nowadays a very upcoming drug called as ceftazidime avibactam is also used. In the case of carbapenem resistant enterobacterials, okay, you can the options which are available again are tigecycline, colistin, aminoglycosides, ceftazidime avibactam. Ceftazidime avibactam act only for KPC and OXA48. You can't use it for, okay. So if you need, then you should add astriol. So that will be that can be noted when you do a prescription audit. In the case of crab, okay, carbapenem resistant Acinetobacter, you will see that the prescription will be for digicycline, colistin, okay, aminoglycosides, sulbactam, etc. In the case of salmonella, generally ceftriaxone and azithromycin will be. The upcoming antibiotics are there in the market. Okay, though ceftazidime avibactam, I can't say it as upcoming. It's already being used. Okay, so ceftazidime avibactam is used for a number of things. Like for example, if they find that the bacteria is having KPC enzyme, OXA48 enzyme. Okay, it is used for enterobacterial, for Pseudomonas, Acinetobacter, etc. The other drugs which are not commonly seen in the market are ceftazolone tazobactam, meropenem barbobactam, meropenem celastin combination with relibactam, bacterocol. Okay, it is a higher generation cephalosporin, plasmomycin, ervacillin. So all these things are there in the market, are available in the market, but in limited amount. Okay, and they're extremely expensive. So you must think twice before you prescribe these drugs to the. I mean, before these drugs are prescribed to the antibiotic to the patient. I'm sorry. So coming to the therapeutic scope in Indian context, okay. So the drugs which are seen these days, okay, in the prescriptions are the carbapenem sparing options. So in the carbapenem sparing options, you will see cefepime tazobactam, you will see cefiderocol, you will see astrionam avibactam combination being used for carbapenem resistant Klebsiella pneumoniae. Okay, you will also see cefepime zidibactam. So these drugs are seen these days in the market. They're also seen in the various prescriptions. Now, moving to antifungal agents. I hope the antibacterial resistant mechanism was clear. Now, we are going to move into antifungal resistant mechanisms. Now, in antifungal resistant mechanisms, before we start talking about the resistance, let us first see how many types of drugs are available for acting against the fungi. So, the first group is antibiotic. Okay, so under this group you have amphotericin and hamycin, which comes under the polyene group. Okay, so in this your amphotericin is very commonly used. Then you have a heterocycline benzofuran, that is your griseofulvin. Okay, and then the next group also your echinocandins, that is the caspofungin and micafuntin, again which is given in the ICU setup. The next group is the azole group, the most commonly used group. Under which you have the triazole, that is the fluconazole is the most commonly used drug there. So fluconazole, voriconazole, itraconazole, and posaconazole. Then you have the imidazole group, where you have your topical antifungal agents like clotrimazole, okay, miconazole, and also your systemic agent like your ketoconazole. Next is your topical, the only topical antifungals which are used like your tolnaftate, okay, cyclopirox, etc., and also terbinafine, that is an allylamine. So these are the various antifungal drugs which are available in the market. Now coming to antifungal resistant mechanisms, I have taken three commonly used antifungals. One is your azoles, okay, then your polyenes, and then your echinocandins, okay. So this particular diagram shows you the resistance caused by azoles in various types of fungi. The first part is for Candida, then for Cryptococcus and Aspergillus. We have concentrated on these three, okay, in today's presentation. So whenever you talk about antifungals, just like bacteria, they also have few common mechanisms. Now, one is the target alteration, okay, the modification of the target site, okay. The second thing is having upregulation of your transporters, okay, multi-drug transporters. So they also have transporters which will transport the drug to and fro into the fungi and outside the fungi. The third thing what they have, which is slightly something different from bacteria, is stress responses. Okay, so that also affects the antifungal susceptibility. Now let us go for Candida. Okay, in the case of Candida albicans, now you will see that mutations, okay, mutations in ergostrol 11, okay, and UPC2, and having a loss of heterozygosity. So these affects the target of the azoles, and they prevent the entry of azoles into the fungi. The second thing is your multi-drug transporters. So here, overexpression of certain multi-drug transporters like CDR1, CDR2, MDR1, mutations, okay, mutations presence, and loss of heterozygosity. Again, this affects the multi-drug transporters, okay. The third thing is ERG3 mutations. Now, they cause stress response dependent resistance. Okay, so because of this, again, your azoles will not be able to act on Candida albicans. Now, coming to Cryptococcus neoformans, which causes meningitis most commonly, it also has systemic cryptococcuses. Here also, mutations in ergostrol 11, overexpression of ergostrol 11. Okay, all these things will affect target of Cryptococcus of the azole antifungals. And also in the case of upregulation of multi-drug transporters, your overexpression of AFR1. Okay, this can also cause it can cause alteration in your multi-drug transporters and prevent the entry of azole into Cryptococcus. The third thing is your stress response mediators. Okay, so when the stress response mediator is affected, again, antifungals, that is your azoles, will not on the Aspergillus, the mutations are slightly different. Here it is the GY54 mutation, which is present in this particular area, the CYP51A. Now, overexpression of certain transporters and HSP90, that is the main mediator of stress response in Aspergillus. So, when that gets affected, azoles will not work on Aspergillus. Okay. So, this is three main by cause azole resistance is caused in fungi. The next is polyene. So in polyene, the most important antifungal we had was amphotericin. Okay. So in polyene, we are going to see only about Candida. So in Candida, what happens is you have to forget about the transporters. Okay. Here only your alteration of the target site and the stress response is involved. So for alteration of the target site, loss of certain loss of function because of certain mutations, okay, in the ergostrol biosynthesis, that causes the resistance. And in the case of stress response, your regulators such as HSP90, when they are affected, okay, they can cause resistance towards polyenes. Okay, so these two things, that is your target site alteration and the stress response mediators, they are responsible for polyene resistance in Candida. Coming to echinocandin resistance. In echinocandin only you have caspofungin, micafungin, etc. So in echinocandins, again, we are going to see in Candida and in mold, that is the Aspergillus. Again, in this, you have to think only about the target site alteration or overexpression of the target site. The second thing is about the stress response. In the case of Candida albicans, it is mutation in the FKS1 gene. Okay. In the case of Aspergillus, again, it is mutations in FKS1 gene, okay, which causes resistance to echinocandin. In the case of cellular stress response, the mediators involved in the case of Candida, again, it's HSP90, along with other mediators like calcineurin. In the case of Aspergillus, the mutations is in HSP90, calcineurin, RAS, etc. Okay, so these causes various changes in the cells which will prevent echinocandin function on antifungal. Okay, so this is a nutshell about all three types of antifungal resistance mechanisms seen in fungi. So that is the end of session. So these are just if there are any, I'll be most willing to address. Okay, thank you so much for your patient listening. Over to you, Vrinda.
Thank you so much, ma'am, for giving a very concise and, like you said, in a nutshell, to talk about such a vast topic. And for me, it was also interesting to see that you also touched upon the clinical relevance of these topics. So many a times what happens is we learn about the theoretical aspect, but we fail to correlate it with, when we come to a clinical practice. So thank you so much for doing that. So I have actually a few points that I actually want to discuss with you based on your presentation.
Yeah.
So
Sure.
you touched upon acquiring resistance from the environment. So there's a lot of papers that talk about how environment plays a role in antimicrobial resistance. So could you just elaborate on that? How do we get resistance from the environment? You mentioned about how it can enter through the food that we eat. So could you just elaborate on that, ma'am?
Yeah, sure. So that is the most, I think, a favorite topic of everybody these days: environmental entry of multi-drug resistant pathogens. Now, we should always have healthy eating habits. Okay, so we know that drugs are present in many of the food items we are having. For example, the most common is having colistin injections for chickens for them to multiply easier. Okay, and having certain metals present in fish. Okay.
Yep.
And very recently, I saw an article wherein even we thought mutton is free of antibiotics, and recently there were articles saying that you are going to have a broiler variety of muttons available. So these days, antibiotics are used for having healthy poultry and for easy access to the poultry. So we must be very cautious in how we are choosing our food items these days. Okay, and it is not only for non-vegetarians, even for vegetarians for that matter. All our food items are being produced with a lot of chemicals and with a lot of insecticides. So many a times these agents are also antifungal agents. Okay, so when we are exposed to these agents through our food, basically our gut environment, the gut bacteria what we are having, it is developing resistance. So many a times what we see is even neonates or even small children, if you see their stool sample and do a surveillance, they will have multi-drug resistant enterobacterials in their stool, which is very surprising because of their age.
Yeah.
Okay, so it's mainly the dietary component which is affecting them. So that health for all issue must be addressed. It is what to say, it's not something to be addressed might be in the distant future. It has to be addressed immediately so that we can stop the spread of antimicrobial resistance starting from our home, not just in the hospital.
Right. Since you are also an IPC expert, I actually wanted to also touch upon the IPC practices that needs to be followed when you have a patient with, say, multi-drug resistant or XTR organism. So what are some of the, especially when the patient is in ICU or in a ward, what are some of the minimal IPC practices that needs to be considered when we have a multi-drug resistant organism in a patient?
Sure. So basically, as you know, there are different types of resistant infections, right? So each has its own type of infection control practices, which we advise clinicians so that they don't spread it amongst themselves. But the most important factor, as we all know, is hand hygiene. That is the most underrated infection control
Yep.
practice, but it is the most useful one. Okay. So the first thing, if you see in every infection, be it MRSA or be it Clostridium, or anything, even for TB for that matter, though TB is a respiratory thing, there could be other infections in that patient. So, hand hygiene is the most important one, no matter what infection it is. And the number two is small things like when we think about hand hygiene, we always think that it is after touching the patient. There are
Hmm.
various moments in hand
Yes.
hygiene. Yeah. So before touching and after touching, even the most important thing is touching the patient's surrounding. So which we have to emphasize our clinical interns and everybody who approaches a patient. So that is very important. The second thing is the other areas which the patient uses. For example, his what to say, the sanitary area, the toilet, what he uses. Okay, so all those has to be clean. The environment has to be clean. And for certain infections, for example, in the case of TB or in the case of say any other like varicella, so it is better that the person is in a negative pressure room. So those things are selective for certain infections. But if you see universally, the most important thing is to have a good hand hygiene practice, to have a proper PPE, appropriate PPE. For example, for MRSA, it is just gloves and your gown. Whereas if you're going to a TB patient, your mask is very important there.
Right.
Your more than anything, a mask is important. So the first thing is hand hygiene. The second thing is appropriate infection control measures has to be taken, so that we as practitioners, we don't get the infection in our hands or in on our body, and we don't spread it to others. So that's the main thing.
Right. So I think I also would like to just re-emphasize here that always antimicrobial stewardship and infection control should go hand in hand. Many a times when we
Definitely.
focus on one, we lose focus on the other part. I think it has to be always taken hand in hand. And you also mentioned about the various factors that is resulting in AMR, and you mentioned about overprescription of antibiotics. But I actually wanted to ask you about even the factor that many patients don't complete their course of antibiotics, and that can also lead to resistance. So you may not see it in a hospital setting because they are being monitored, but when you go to the community setting, I think that's another major factor that's leading to resistance in the long run. So how do they? In that case, what is like the mechanism of resistance that's occurring there? Like if the patient is not completing the course.
Okay, so again, as you said, it's very common. So even being medicos, sometimes we forget to, complete the course for our children itself, like when they have certain infection. So what happens here is, it is not intrinsic resistance here; it is acquired resistance.
Yes.
So the bacteria is sensitive to your antibiotic. Your bacteria is getting killed. You are seeing some improvement in your child or whoever is in the house, and that is when you forget about giving the next dose. So it is something, it's an susceptible antibiotic which your child or somebody was having in your family. Now, when you stop the course midway, what happens is there are certain, what to say, a certain, like for example, there were 100 bacteria in your body. Not everything would have got killed. You would have few bacteria which are still persisting. Now, you have not completed your course. Hence, you have not completed killing the entire microbial load which is present in your body. So they are still present over there. And repeatedly, when you keep doing this, what happens is these particular bacteria will develop certain mutations. It can develop either mutations or, for example, if it was a resistant bacteria, you might actually spread it to another person also.
Yeah.
Okay. So your body is now developing resistance mechanism. So it can be anything. It could be mutation. It could be a transferable resistance, like a plasmid resistance you have in your like certain bacterial cells will be resistant. Or 100 bacteria need an. So out of your 100, a few resistant bacteria. I hope I did not lose your signal. Okay. So, sorry, I got a call in between. So, out of your 100, not all 100 will be sensitive. So, at least few will be resistant. Okay. And it is very important to finish your course so that you eliminate your entire microbial load. And if at all you have any resistant strains, you're able to pick up in that particular stage itself, because you will not be responding after that. Okay. So, it is the acquired resistance which develops if you stop your antibiotic midway. You are making your body susceptible for mutational resistance, or for most often it is mutational resistance, not the plasmid mediated. So that is what we cause to our body when we do this stopping of the antibiotic midway, and then when we think that it is not.
Yeah, I think that was very informative, and many times we forget that part. Like we always focus on overprescription, especially from an antimicrobial stewardship point of view. We focus on irrational prescribing behavior, but we forget about the patient aspect, like whether the patient is actually completing the course or not. So that is also very important when you think from a AMR perspective.
Yeah.
So I think, thank you so much, ma'am. It was a very interesting session, and you did complete it in a very crisp and concise manner. So thanks a lot for that. So
Thank you so much. If there are any other questions which your audience would have, I'm very much happy to, answer them. So they can send it to me through email, or I've mentioned my mail ID. So they can send
sure,
it
ma'am.
through email. So I'll be more than happy to reply.
Thank you, ma'am.
Thank you so much, Vrinda.